Immunology Insights, Basics and Beyond. Chapter 19, Phagocytosis. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we explored how the immune system defends against pathogens through the inflammatory response, leveraging various innate and adaptive mechanisms. One of the most fundamental and powerful of these mechanisms is phagocytosis, a process that allows immune cells to engulf and digest invaders. Today, we are diving deeper into phagocytosis. While already touched upon in previous chapters, we now go more in detail as this process is crucial for both innate and adaptive immunity, enabling immune cells to recognize, engulf, and destroy pathogens, cellular debris, and other harmful substances. It forms the first line of defense against infections and plays a significant role in the immune regulation. We will explore the key players in phagocytosis, the molecular mechanisms behind it, and its significance in immunity and disease. So let's talk science. Let's start with the key players in phagocytosis. Phagocytosis is primarily carried out by specialized immune cells known as phagocytes. The most prominent phagocytes include: first, macrophages. These are tissue-resident cells that patrol their environment, engulfing pathogens and presenting antigens to T cells to coordinate an adaptive immune response. Second, we have neutrophils. They are short-lived, highly motile cells that rapidly respond to infections, primarily targeting bacteria and fungi. They are abundant in pus and contribute to inflammation. Third, we have dendritic cells. They act as professional antigen-presenting cells, efficiently capturing and processing antigens to activate naive T cells and bridge innate and adaptive immunity. And fourth, we have the monocytes. They circulate in the bloodstream and differentiate into macrophages or dendritic cells in tissues, contributing to both immune surveillance and tissue repair. Each of these phagocytes plays a unique role in the immune defense, but all share the ability to recognize, engulf, and destroy harmful invaders. Now let's have a closer look at the phagocytosis process. Well, phagocytosis occurs in several well-coordinated steps. First, we have recognition and binding. Phagocytes detect pathogens through pattern recognition receptors or PRRs, which recognize pathogen-associated molecular patterns or PAMPs. Complement proteins and antibodies can also opsonize pathogens, enhancing their recognition by phagocytes. Second, we have the engulfment. The phagocyte extends its membrane around the pathogen, forming a vesicle called phagosome to internalize the pathogen. Third, we have the fusion with the lysosome. The phagosome merges with the lysosome to form a phagolysosome, where digestive enzymes and reactive oxygen species degrade the engulfed material. Fourth, we have degradation and antigen presentation. The pathogen is broken down internally into small fragments, some of which are loaded onto major histocompatibility complex or MHC molecules. To reiterate here the most important details, we have first MHC class I molecules that present intracellular pathogens-derived peptides such as viral antigens, for example, to CD8 positive cytotoxic T cells, leading to the destruction of infected cells. And second, we have MHC class II molecules that present extracellular pathogens derived from peptides to CD4 positive helper T cells, which orchestrate an immune response by activating other immune cells. Such an important process as phagocytosis, of course, has roles in health and disease. Phagocytosis is essential for immune surveillance and tissue homeostasis, but defects in this process can lead to severe consequences. For example, chronic granulomatous disease or CGD, which is a genetic disorder where phagocytes fail to produce reactive oxygen species, leading to recurrent bacterial and fungal infections due to ineffective pathogen clearance. Or tuberculosis, where the Mycobacterium tuberculosis can survive within phagocytes by inhibiting phagosome-lysosome fusion, allowing the bacteria to persist within macrophages and evade immune destruction. And of course, cancer, Some tumor cells evade phagocytosis by expressing "don't eat me" signals, such as CD47, preventing immune clearance and allowing tumor growth to continue unchecked. Conversely, excessive or uncontrolled phagocytosis can contribute to autoimmune diseases and chronic inflammation. A concrete example is hemophagocytic lymphohistiocytosis, or HLH, where overactive macrophages engulf blood cells excessively, leading to severe immune dysregulation and organ damage. There is, of course, also therapeutic exploitation of phagocytosis. Phagocytosis is a promising target in immunotherapy, with applications in cancer, infections, and autoimmune diseases. For example, in cancer therapy, where tumors evade phagocytosis by expressing CD47, a "don't eat me" signal. Anti-CD47 antibodies restore macrophage function, promoting tumor clearance. Or in infectious disease treatment, some pathogens, like the Mycobacterium tuberculosis, block phagosome-lysosome fusion. Interferon gamma, or IFN gamma, enhances phagosome maturation, improving bacterial clearance by stimulating macrophages. Or in autoimmune disease management, in conditions like hemophagocytic lymphohistiocytosis or HLH, excessive phagocytosis destroys healthy cells. Immunosuppressants help control overactive macrophages. By modulating phagocytosis, therapies aim to boost immune defenses against cancer and infections or reduce immune overactivity in autoimmune conditions. To conclude, phagocytosis is a powerful mechanism of host defense, critical for pathogen clearance, immune regulation, and antigen presentation. It not only eliminates harmful invaders but also plays a crucial role in shaping adaptive immune responses by presenting antigens to T cells. For example, macrophages clear apoptotic cells to prevent tissue damage, while dendritic cells activate T cells against pathogens. Understanding this process helps us develop therapies targeting infections, cancer, and immune disorders. In the next chapter, we will discuss antigen presentation, a process closely linked to phagocytosis that bridges innate and adaptive immunity. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So, see you next time, and thank you for listening.